Hey, welcome to Chapter 41, Animal Nutrition. By the end of this chapter, you should be able to identify the major groups of nutrients that animals require to complete their life cycle. You should be able to discuss the structure and function of each digestive tract organ in the digestion and absorption of nutrients. You should be able to describe the causes, symptoms, and consequences of type 2 diabetes, and that's all. So we're not going to be covering too much in this chapter. All right, so animals have two basic requirements that they must have. They have to be able to obtain the chemical energy in order to make ATP to power their cells. And animals also need to be able to get carbon-containing compounds to be able to build new molecules. So animals get energy from food by ingestion, meaning bringing food into the digestive tract, which is just eating things. Digestion, which is breaking down food into small enough pieces to allow the nutrients to be extracted. Absorption, the uptake of the specific ions and molecules into the digestive tract. And then finally, elimination. That's just getting rid of the waste. Okay, so we talked about essential nutrients a little bit in the plant chapter, but essential nutrients are just nutrients that can't be synthesized and have to be obtained from diet. So some amino acids are essential, some we can make ourselves. Some fatty acids we need to be able to get from other organisms. We need to get vitamins from eating things. So vitamins are organic compounds that we need for health, but we only need small amounts of. And then minerals are inorganic substances, and so they're used for different things. Some of them are enzyme cofactors. Does anyone remember what that is?
Yeah, a cofactor is like a mineral that gets in there in the enzyme in the active site and makes it fit the substrate.
Yep, that's a cofactor. They can be part of our structural materials, and also the ions from minerals are the major electrolytes in the body. Okay, so which of the following is not an essential nutrient in humans? All right, A, fatty acids called essential fatty acids that must be obtained from eating certain plants or fish. B, all 20 amino acids required to manufacture most proteins. C, vitamins, organic or carbon-containing compounds that are required in only minute amounts. Or D, minerals that are inorganic substances used as components of enzyme cofactors or structural materials. All right, let's go through them. Well, can anyone eliminate any of them?
Yeah, well, vitamins are essential nutrients, so that's not it.
Okay, what else?
Minerals are essential, so that's not it.
Okay, what else?
Well, some fatty acids are essential. I guess essential fatty acids are essential, right? Okay, but then some fatty acids you can make yourself.
Yeah. What about B, all 20 amino acids required to manufacture most proteins?
I think that's the answer because, we can make some of those amino acids. We just can't make all 20 of them.
Right. Okay. So not all 20 amino acids are essential. Some we can make ourselves, but some of those are essential. So that statement is not correct. All right, let's start with the mouth and the esophagus. So digestion begins in the mouth. So within the mouth, there is salivary amylase. So what kind of a molecule is that?
It's an enzyme.
Yeah, how do you know it's an enzyme?
Because it ends in ASE.
Yes. So if it's called salivary amylase, what do you know about it?
Oh, it's in the saliva.
It's in the saliva, yep. And it is also involved in breaking down the bonds between glucose molecules, right? So starch is made up of a whole bunch of glucose molecules. Maltose is just 2 glucose, right? So salivary amylase is breaking the bonds to release little maltoses from longer chains of starch or glycogen. And so that initiates the digestion of these molecules. And then cells in the tongue will synthesize and secrete lingual lipase. So what do you think that one does? Well, lingual means like tongue. Yeah. And what do you think a lipase breaks down?
Oh, it's lipids.
Yeah, lipids, right? So lingual lipase begins digestion of lipids, so it starts breaking down triglycerides into diglycerides and fatty acids. So do you remember from 1113, what's a triglyceride?
Oh, that's a fat where it's like a glycerol and 3 fatty acids.
Yep. So that's a triglyceride. So that will get broken down into a diglyceride and fatty acids. In the mouth are salivary glands, and they make mucus. And so the combination of water and mucus has to be there in order for the food to be swallowed. Otherwise, it would get stuck in your throat. There is a tube called an esophagus that connects the mouth to the stomach. And so the food is propelled to the stomach by a wave of muscular contractions called peristalsis. Okay, so it's the same kind of motion, for example, that a worm makes when it moves. And the kind of muscular contraction that results in swallowing that results in the food being propelled to the stomach is stimulated when you swallow. So you have a muscle contraction that constricts the esophagus above the food, and then it moves like a wave down the esophagus, pushing the food below it into the stomach. All right, so the stomach here is a tough muscular pouch. It has these ring-like kind of cinches called sphincters that control the passage of food into the stomach and out of the stomach. And so the muscles in the stomach contract, and when that happens, it physically just churns up and breaks down the food in that way. And the stomach also is able to partially digest proteins as well. So the lumen of the stomach, have you heard of the term lumen before? Yeah, I remember the lumen. It's like the area inside of a chloroplast. That is true. So usually when we mean lumen, it's just like a space inside of a thing. So the lumen of the stomach is just the inside of the stomach, right? And that neighborhood is very acidic with hydrochloric acid, HCl. So some of the early work on the stomach was done by Dr. Beaumont. And so he treated a Civil War gunshot victim who he got shot in such a way that he had a hole in his stomach, but then it healed up, leaving a kind of a window in his stomach to the outside of his body. So you could actually look into the hole and see his stomach kind of and have things churning. So Dr. Beaumont would take little pieces of food on string and stick them in the hole in the guy's stomach and watch the process of digestion. So the enzyme responsible for digesting proteins is called pepsin. And so the stomach cells contain not pepsin itself, but pepsinogen, which is a pepsin precursor. Okay. And then when the pepsinogen is placed in hydrochloric acid, the pepsinogen is converted to pepsin. All right, so why do you think that the stomach cells produce a precursor to pepsin rather than producing pepsin directly?
Wow, it is extra complicated. Why not just make pepsin?
Well, why not just make pepsin? What does pepsin do?
It breaks down, proteins and other foods.
Okay. So if the cells made the substance that broke down proteins and other foods, what would happen to the cell?
Oh, the cell will get digested. That's so gross.
Okay, yeah. So the stomach cells have to produce the precursor to pepsin, pepsinogen, and then put that in the hydrochloric acid so that it can get converted to pepsin outside of the cells. Again, so that prevents destruction of proteins in the cells where the enzyme is actually made. Okay, the material then moves by peristalsis through the stomach sphincter into the small intestine, which is basically like a long folded tube here. And the food will mix with secretions that are coming from the pancreas and liver. Oh, let's look at the structure of the small intestine. So if you look in cross-section, you can see it has a lot of infoldings. And then if you look closer, each of these infoldings is covered with small finger-like villi, which are caused by folding. Okay, so this is an individual villus. And then if you look in even greater detail, you can see that along each one is a microvilli, like this. So you have lots and lots and lots of surface area available. And so this can absorb the little bits of food and nutrients that have been released. And so by the time the food gets to the end of the small intestine, digestion is complete. So most of the nutrients, along with most of the water, have been absorbed by the cells. And so the small intestine, as I just said, has a huge amount of surface area with villi and their tiny little projections called microvilli. And so because there's so much surface area, it increases the rate at which nutrients can be absorbed. So we're very efficient at removing all of the usable nutrients from the food that we eat. All right, by the time the digested material has gone all the way through the large intestine to the from the small intestine to the large intestine, almost all the water and available nutrients have already been absorbed out of it. So the primary function of the colon is to be able to form feces and absorb any additional water and compact the wastes that are left over. So the feces are held in the rectum until they can be eliminated. Another kind of cool thing that the human colon can do is that it has this symbiotic colony of microorganisms that help us digest cellulose. We're not super good at doing that, so it's kind of one of these things where, like, a cow, for example, has really good kind of little colony of microorganisms to digest cellulose. We don't have like that kind of an operation going on. We can kind of digest a little bit of the cellulose, but not very much. But those bacteria are really important because they help produce nutrients like vitamin K that we would be unable to make ourselves. So we kind of just keep a little like farm of bacteria in our body to help us make vitamin K and to digest a little cellulose. All right, so here's a picture of the digestive tract. All right, so we're starting with the mouth, so that's where the food is going in. It's being physically chewed up and chemically processed, right? The salivary glands are secreting enzymes that digest carbohydrates and create lubricating mucus. The esophagus is transporting the food. The liver is secreting molecules that help digest fat, and the liver secretions are going into the stomach, where which is where mechanical processing, like kind of churning, happens. The gallbladder is also secreting. It's storing secretions from the liver and then emptying those into the small intestine. And then the pancreas is secreting enzymes and other materials into the small intestine. So the small intestine is where most of the chemical processing and absorption happens, and so most of the nutrients and water are removed. In the large intestine, the last little bit of water is extracted, and this is also where the feces are formed. We also have symbiotic bacteria in the large intestine. We consider the appendix. Part of the digestive tract is kind of attached off of the large intestine, and it contains some immune tissue which harbors symbiotic bacteria as well. Finally, at the anus, waste is eliminated. All right, so let's put the organs in digestion order. So first, match the substance to the organ where it can be found, and then after, we'll figure out what they do. So let's start at the beginning. If we put them in digestion order, okay, so just look at the left-hand column. What's the first one?
It's the mouth.
Yeah, the mouth. Then what? The esophagus. Yeah, the esophagus. Then what?
The stomach.
Yeah.
They forgot the gizzard.
Well, humans don't have a gizzard.
Okay, then this goes to the stomach.
Yeah. Then what?
Small intestine.
Yeah, then what?
Large intestine.
Yes, then what?
Colon and anus.
Okay, good. Let's match up the substances to the organ where they can be found. HCl.
Stomach.
Yeah, that goes in the stomach. Lingual lipase.
That's the mouth.
Yeah. Mucus.
The mouth.
Yeah. Pepsin and pepsinogen.
That's the stomach too.
Yeah. Salivary amylase. That's the mouth. Yeah, and then symbiotic microorganisms.
That's a colon.
All right, so we got those in order. We got this in order. Well, I guess we say the symbiotic microorganisms are in the large intestine, but yeah. Other than that, all right. Oops, we didn't talk about what everything does. Just remember, salivary amylase is breaking down starch and glycogen into maltose. Lingual lipase is breaking down triglycerides, right? Mucus allows the food to be swallowed. HCl helps convert pepsinogen to pepsin. Pepsinogen is converted into pepsin, and then pepsin digests the proteins. And in the large intestine, the symbiotic microorganisms digest cellulose, and they could also produce vitamin K. All right, we're going to come back to talking about nutritional homeostasis later, and we'll go into, if you take biology 3401 with me, we'll talk a lot more about diabetes as well. But let's just think about glucose as a case study here. So after you finish digesting, right, your amino acids, your fatty acids, your ions, and your sugars are in the bloodstream and eventually are delivered to the cells, right? So insulin is a substance produced by the pancreas, which is responsible for controlling the uptake of glucose, okay? So if your blood glucose levels fall too much, then the pancreatic cells will secrete a hormone called glucagon. So this is kind of the process here, okay? So let's just say you have just eaten a bunch of stuff, okay? So you have very high glucose levels, right? You just ate a whole bunch of sugar. Okay, your pancreas will now secrete insulin. Insulin will cause that glucose to get locked up into glycogen, a storage molecule, right? Okay, so now you have less glucose in your body, and you're at homeostasis. So now you have normal glucose levels in the blood, okay? All right, and then maybe it's several hours later, right? And you've used up all the glucose, and you start to have low glucose levels, right? So now your glucose levels have dropped too low. At this point, your pancreas secretes glucagon, and the glucagon will take the glycogen and break it back into glucose again. So now your sugar levels go back up, and you're back in homeostasis, okay? So this is a kind of a feedback system that allows you to always have the correct amount of glucose mobilized in your bloodstream so that you can power your cells. If you have a disorder in this kind of system, it results in diabetes. So diabetes develops in people who either are unable to secrete insulin so that the glucose can't get locked into glycogen. So if you're not secreting, if you're not synthesizing insulin, that's called type 1 diabetes. Or some people, their pancreas is making the insulin, but their body is resistant to it. So the insulin isn't effectively activating the receptor in the target cells to be able to take the glucose and lock it into glycogen. So that is called type 2 diabetes. All right, so hopefully you learned how animals ingest, digest, and absorb nutrients. We talked about essential nutrients, how they're incorporated. We went through the mouth and the digestion, and then we asked how you maintain nutritional homeostasis and used an example of type 2 diabetes as a imbalance in homeostasis.